Data analysis is going to be a breeze, so follow along with Module 1 to learn how to open a run file, verify the fluorescent standards, verify the biotintillated ladder, and name peaks. Ready? Let's get started. To open a run file, open Compass and click on File, then Open, Browse, and navigate to the Compass data file with the .cbz file extension. The default location for runs is in Documents/Compass/Runs, but an alternate save path can be selected when starting a run. A movie playback of the separation can be viewed by clicking the Run Summary icon and the Separation tab. These standards are used to align data between capillaries and help with sizing. The 1 kilodalton standard should always be the strongest or tallest signal, and it is important to assign these standards correctly in each capillary. Compass does a great job of detecting the standards automatically, but once in a while you may need to manually fix a standard. Click on the Analysis icon, then select the Show Standards in Single View icons. In the Graph View tab, check that all the capillaries have the standards identified correctly. If any of the standards are assigned incorrectly, right-click on the correct peak and select Force Standard. Select each capillary in the Experiment tab and ensure alignment similarly. Alternatively, you may also click Not a Standard to deselect a misaligned standard. Similar to the fluorescent standard peaks, the correct assignment of the biotintillated ladder peaks must also be verified. To do so, click on the Show Samples and Single View icons, and then select the ladder capillary in the Experiment tab. In the 12 to 230 kilodalton kit, the biotintillated ladder should show the following sizing peaks: 12, 40, 66, 116, 180, and 230 kilodaltons. When using the high molecular weight kit, the ladder peaks will be 66, 116, 200, 280, and 440 kilodaltons. Make sure these are correctly identified in the Graph View tab. If a peak is incorrectly identified, right-click on it and select Remove Peak. If a ladder peak is not visible because it falls outside of the range of the graph, click View, then View Region, and select Full Range. Peaks in Graph View are labeled automatically with the calculated protein molecular weight size. Fully analyzed results, including molecular weight, peak area, peak height, and signal to noise, are shown in the Peaks table. In Lane View, you can see the calculated molecular weight when you move your mouse over the band of interest. Specific sample peaks can be labeled by clicking on the Edit menu and selecting Analysis, then selecting peak names within the analysis screen. Peak name settings are saved as a group, and multiple settings groups can be created. Specific group settings can then be applied to individual capillaries, primary antibody names, and attributes in the run data. Peak name groups are displayed in the Analysis Settings box. Once a name for the group has been entered, peak names can be added and edited in the Analysis Settings Protein box. Select the first cell in the name column in the Analysis Settings peak table and enter a sample protein name associated with the primary antibody used in the run. Next, select the first cell in the molecular weight column and enter a molecular weight for the sample protein. By default, Compass will automatically name peaks found within 10% of the molecular weight entered, but this range can be edited in the Range column. The custom color selected in the Color column will be used to identify the sample protein peak in the Peaks and Capillaries table in the Analysis screen. To add another sample protein to be labeled in the same capillaries, click Add under the Analysis Settings peak table and repeat the previous steps. The final step in peak naming is to apply the peak name groups to the appropriate capillaries within the run data. In the Apply Settings box, click the cell in the Apply To column. Then click the down arrow and select an option from the drop-down list. Analysis settings groups can be applied to all capillaries, to specific capillaries according to primary antibody names or attributes, or to a custom list of capillaries. Click OK to save changes and exit. The bands and peaks in the sample are now labeled with your peak names. To learn how to see your data in different views and make figures, continue to Module 2.